Alright, let's get prepped for tomorrow's biology test. First of all, who is the father of genetics? His name is Gregor Mendel. He's an Austrian monk, and we know him as the father of genetics because he worked out probability ratios so that he could predict what the offspring would look like from certain crosses, and his work was all with peas, which probably was very boring. He came up with a word called allele. An allele is a variation of a trait. So, for example, if I've got a trait like eye color, my variations, or my alleles, would be brown, blue, green, hazel, etc. Or maybe I'm talking about, let's say, hair color in horses. So maybe I've got some brown horses and black horses and gray horses, etc. Those are all alleles. Where do we get each of our alleles from? One from each parent. That's why you have two copies of everything. Since you have one from each parent, we call that the law of segregation. And that's not segregation like in history class, that's segregation that means that those alleles are split. Clearly one comes from dad and one comes from mom. Let's go through some vocab next. What is the difference between dominant and recessive? Dominant, we're always going to abbreviate with a capital letter. Recessive, we'll always abbreviate with a lowercase letter. Dominant, you only need one copy of that allele, and then you'll see it. Recessive alleles are like hidden alleles. You need two copies of the allele to see it. So remember my example from Pitch Perfect. This is the really loud, noisy singer. You hear them no matter what. This is the super quiet singer. You only hear them if they're the only person in the room. Homozygous and heterozygous. The difference here, homo means same, hetero means different. So a homozygous individual is someone who has two copies of the same allele. So either two big A's or two little a's. So two copies of the same allele. Running out of room. Heterozygous means different, so this would be big A, little a. Two different versions. Big A, little a. Next vocab terms that sometimes get confusing are genotype and phenotype. So genotype and phenotype, the difference is genotype is what your genes or your alleles say. So this would be, for example, big A, little a, big A, big A. That's your genotype. That's what we know is on your genetics. Your phenotype is what you look like. So this is your physical appearance. A good way to remember that is simply how these words start. Genotype, what your genes say. Phenotype, physical appearance. What do you actually look like based on your genes? Let's do some Punnett squares. So this is an example of simple dominance or complete dominance. And in this problem, it says that SpongeBob is determined by the allele Y, yellow is dominant, blue is recessive. So that immediately tells me recessive, lowercase letters. So I'm going to put a little letter Y, lowercase y, next to that. Next, it says that SpongeBob and his mate are both heterozygous. Well, we just learned heterozygous means one of each. So that's a big Y, little y. So remember, if it's dominant, you only need one copy of the capital Y to look yellow. So here I've got 1, 2, 3 of them that are going to look yellow. 3 out of 4 equals 75%. That leaves us with this little guy right here, little y, little y. We're going to only see that that homozygous recessive, which is our blue. 25%. Incomplete dominance, remember that this means that they're going to blend, they're going to mix. Neither of them is strong enough to be the only one seen. So this is like when you mix paint. So here we've got blue and pink make purple. Squidward is blue, so let's go ahead and put him on one side of our Punnett square. And he has a purple partner. So let's fill in the Punnett square. This one, blue, blue. This one, blue, pink, so that looks purple. This one, blue, blue. This one, blue, pink, looks purple. So if I look here, these two are blue. That's 50%. I don't have any pink, because remember these would be two lowercase. I do have two purple. So 50% blue, 50% purple in that case. Hopefully you guys can see that. Next example is codominance. And codominance means that you're going to see both. Like they're cooperating, they're side by side. Here, we're going to always use the example for blood types, just because it makes the most sense. So it says she is heterozygous for blood type B, and then it gives you the genotype here. So I'm going to draw my Punnett square, and she's I, the name tag, with a little b attached, and then this little I, which means no name tag at all. And then it tells us that Randy the squirrel is heterozygous for blood type A, and this is his genotype. So what I look at here, this one has two name tags. They say A, B. That tells me that we've got 25% of our offspring are going to be type AB blood. And we're back, so let's do the rest of this problem. So we figured out that IA, IB meant that 25% of these kids are going to have AB blood type. Then we've got this one right over here, and this one is IBI. That means that they've got a name tag that says B, and then no second name tag. So that individual is going to be type B blood, 25%. This individual, same way, they only have one name tag, and it says A on it, so they're type A blood. And then this one has no name tags, they're just blank, so they are type O blood. So 25, 25, 25, 25. You will always see that if both parents are heterozygous. X-linked, remember this one, it matters the gender of the individual. And remember that boys are XY and girls are XX with those X chromosomes. So we're going to have this allele for intelligence, and it says that Patrick is not intelligent, so that's X with a little I attached. What is Patrick's other chromosome? Simple. He's a boy, he's got an Y. I'm trying to write and talk. So XI, that's Patrick. And then Patricia, I don't know why I chose these names. Patricia, it says, is a carrier. Now that means that she doesn't have that, but she could pass it on. So she's X, big I, X, little I. What is the probability that their daughters will be intelligent and their sons will be intelligent? Let's fill out the square. Here's a girl, she's got two X chromosomes, and she's big I, little I. Here's a girl, two little I chromosomes. Here's a guy, and here's the other guy. So it says here, this daughter has two copies of the not intelligent gene. So she's not intelligent. This one is. So of their daughters, 50% will not be intelligent. Now let's look at their sons. This one is not. So 50% again. Dihybrid crosses, remember, are the gigantic ones where we build the giant squares. So I'll show that two ways. I'll show you how to set up the 4x4 square. I'm definitely not going to fill it all in. And I'm also going to show you how to do the multiplying probabilities. At least I'll get you started on that. So it says SpongeBob is giggly and annoying. So his genotype is big G, big A, little a. And his wife is the exact same, big G, but little a. Okay, so the very first step that we're going to do for a dihybrid cross, if we're going to do the big 4x4 square, is that FOIL method. So this big G could go with this big A, this big G could go with this little G, big A, little G, little a. I'm going to need to fill those in. I have two big G's, a big A, and a little a. And so on and so forth. You would fill that out. But there is an easier way, and this is the way actually that I prefer. I like multiplying probabilities because this gets messy. So instead what I'll do is I'll set up two Punnett squares. One for the giggly thing, and that's the giggly thing. So we've got 75% giggly and 25% that are not. And then our second box, SpongeBob is annoying, but his wife is not. So we've got 50% annoying and 50% that are not. So what we do for here is if we're looking for giggly and annoying, we simply multiply 75% times 50%. 0.375. Giggly, but not annoying, 75 times 50%. Same thing. Not giggly, that's 25%. Annoying, 50%. So 25 times 50% is 0.125. Not giggly, not annoying. Same thing, 25% times 50%, 0.125.